Hi class, let's start this section with a Mplus exercise. This was a study about stability of feelings of alienation over time by Winton and colleagues. Data were collected on feelings about anomia and powerlessness in 1967 and 1971 from a sample of 932 participants. I provided the covariance matrix for you to conduct a two-step latent factor SEM analysis. Please pause the video right here, complete both the measurement and structural models before you proceed. Now, let's take a look at the command file I created. First, I tell the program to read a data file that contains a covariance matrix based on a sample of 932. Then I provide the variable names. In the model section, I have the statement to specify factor-indicator relationships. There are 3 latent factors, each with 2 indicators. By default, the first indicator is the reference variable for that factor. In the last block, I request the output information and save the file and run the analysis. If you did everything correctly, the fit indices of the initial measurement model needs improvement in terms of the model chi-square value and the RMSEA. Based on the model fit indices, the only change you need to make is to add an error covariance between anomia 67 and anomia 71. This change is conceptually acceptable because it's for the same measure taken 4 years apart. The error covariance was caused by the common method. Add this change to your command file, save the change, and run your analysis. Now you have a measurement model with good fit indices. You can do the second step by adding the structural relationships to the command file. Don't be surprised when you see the same model fit indices in step 2. It's expected because you have a saturated model in which all pairs of the factors are connected. In other words, your structural model is equivalent to your final measurement model. That's why your final measurement model and the structural model share identical fit indices. At this point, we need to be clear that the purpose of the two-step process is to isolate the data model fit associated with the latent factor structural effects in step 2 from that associated with the measurement portion of the model in step 1. The important issue here is the quality of the data collection. If you use good measurement to accurately capture the information of the latent factors, your model will allow potential structural misspecification to pass fairly freely into the observed data, and you will be able to identify structural misspecification if there are any. However, if the data collection were done using method or instrument that are inappropriate for the underlying construct, the poor measurement can create a lot of noise. As a result, the data are insensitive of variations in latent factors and prevent you from detecting the effect of structural misspecification. Poor quality measurement can have impact in several aspects. For example, you may have better looking overall data model fit indices than what really happened in your model. When you check the modification indices, you would not be able to identify misspecified structural parameters because the poor data quality would give you smaller chi-square differences. Similarly, when you compare the final measurement model to the structural model, you may end up with a smaller chi-square difference as well. Also, the estimate structural parameter may have larger standard errors when the quality of measurement is poor. The takeaway here is that The measurement model acts as a conduit for structural misspecification to pass into the observed data. In order for you to evaluate the merit of the structural relationships, you need to first make sure the quality of measurement during the data collection stage. One way to verify the quality of measurement in SEM is to supplement the two-step analysis with a pseudo-measured variable path analysis. The idea is to get the saturated factor covariance matrix from the measurement step in the Mplus output file, treat it as the input file, and do a measured variable path analysis. And compare the model fit of the pseudo path model with those of the two-step latent factor model. Here is the command file of the measurement model for the exercise behavior example. I added tag 4 in the output section of the command file, which produces the covariance matrix for the latent factors in the output file. I can copy the covariance matrix and save it in the data file. Now I can write a command file to read that covariance matrix and treat the latent factors as measured variables in this analysis. The other information stays the same. Once I run the analysis, you can see here in the output file of the pseudo path analysis, the model fit indices. The chi-square has a value of 174.34, which is statistically significant. RMSEA is 0.242, CFIA is 0.93, and the SRMR is 0.114. The fit indices are much worse than those of the two-step latent factor analysis. This is an indication that the data quality was poor and the theory may not be as good as it appeared in that study. Another topic is about partially latent SEM model. The two studies we have seen so far are both fully latent SEM models because all factors in the structural models are latent and measured by multiple variables as indicators. In partially latent models, there is a combination of latent factors and measured variables in the structural relationships. In Mplus, we treat partially latent models the same as fully latent factor models and consider the measured variables as latent factors with single indicators. For factors with single indicators, we need to estimate the measurement error variance of the single indicators by either finding information from previous studies or setting the error variance to 0. In this diagram, I added gender to the alienation example. Since gender is a measured variable, this model now is a partially latent SEM model. To analyze that partially latent model in Mplus, I created a latent factor called L-gender with only one indicator and set the error variance of that one indicator gender to 0. The last topic is about Haywood cases. When you run SEM with real data set, sooner or later you will encounter error messages in the output file such as covariance matrix not positive definite. It is an example of Haywood case. Variances as squared values of standard deviations must be positive. Negative variances are illegal values known as Haywood cases. Such illegal values may suggest that you have problems in the data structure, model specification, or identification. There are no fixed solutions when you have Haywood cases. You need to check factor intercorrelations for evidence of discriminant validity or multicollinearity. Remember, factors are expected to have low to moderate correlations, Strong correlations between them may indicate too much overlap.